Hello, curious chemists. Did you know proteins can be like the wardrobe of your favorite superhero, different costumes for different missions? Imagine sorting your clothes into simple t-shirts, fancy jackets, and those tricky hybrids that change form. Well, proteins classify similarly based on their chemical makeup and structure into three types: simple, conjugated, and derived proteins. Simple proteins are the easiest to understand. When broken down by hydrolysis, they yield only amino acids, kind of like taking apart a brick wall to just bricks. Classic examples are albumins and globulins, which you might find hanging out in your blood plasma. Next up, conjugated proteins are a bit more complex. They carry a non-protein part called a prosthetic group, like a metal ion or lipid, almost like having a superhero with a gadget sidekick. Hemoglobin is the star example here, with iron at its core helping to transport oxygen in your blood. Finally, derived proteins are like the leftovers from a meal. They come from partial hydrolysis of either simple or conjugated proteins using acids, alkalis, or enzymes. Think of them as fragments or modified versions rather than the full protein. Picture, if you will, a flowchart branching out from proteins into these three groups, each decorated with examples to make sense of this classification. Proteins everywhere, from your muscles to your meals, fit neatly into these categories, showing just how versatile they are. Hey science detectives! If proteins were buildings, they'd be skyscrapers with multiple floors, each level adding more detail and function. Proteins have 4 levels of structural organization: primary, secondary, tertiary, and quaternary. Let's start at the ground floor. The primary structure is simply the linear sequence of amino acids connected by peptide bonds, like beads on a string, each bead a different flavor depending on the amino acid. Moving up, the secondary structure is where the string folds into neat patterns, commonly the alpha helix, which looks like a coil spring, or the beta-pleated sheep that resembles a folded fan. These shapes are stabilized by hydrogen bonds, kind of like Velcro holding fabric folds in place. Climbing higher, the tertiary structure is the full three-dimensional folding of this coiled or folded chain, held together by interactions like disulfide bridges and hydrophobic side chain attractions. Imagine the protein folding tightly like a complex origami. Lastly, some proteins reach the penthouse with quaternary structure, when multiple folded polypeptide chains come together into one big functional unit. Picture a hierarchy diagram showing these 4 levels stacked and connected, helping you see how a simple chain becomes a sophisticated molecular machine. This layered structure is what lets proteins carry out truly amazing biological tasks. Greetings, molecular builders! At the heart of every protein are amino acids, organic compounds that feature both an amino group containing nitrogen, written as -NH2, and a carboxyl group, -COOH. There are 20 standard alpha amino acids that differ by their unique side chains, called R groups, giving each amino acid its distinct personality. Now, here's the chemistry magic. When the carboxyl group of one amino acid meets the amino group of another one, they join hands by forming a peptide bond and release a molecule of water in the process. This peptide bond is a strong covalent link, specifically a -CO-NH- connection that ties amino acids into chains called dipeptides, tripeptides, or, for the big guys, polypeptide chains. Imagine a reaction scheme in your mind showing 2 amino acids coming together, water molecule parting ways, and a new bond locking them in a protein dance. This elegant process is how life builds its protein toolkit, turning simple building blocks into complex, functional molecules. Hello, life enthusiasts! Proteins are the multitaskers of the living world, performing roles from chemical reactions to structural support. They act as enzymes, those biological catalysts like ureas that speed up reactions, hormones like insulin that regulate vital processes, and antibodies such as immunoglobulins that defend the body against invaders. On the structural side, proteins build tough materials, keratin toughens hair and nails, collagen strengthens connective tissues, and actin with myosin powers muscle fibers, enabling movement. Transport proteins play a crucial role too. Hemoglobin famously carries oxygen through your blood, while albumin keeps your blood's osmotic pressure in balance, ensuring that fluids stay where they should. Imagine a chart mapping types of proteins to their functions and examples: enzymes, hormones, antibodies, structural and transport proteins, all neatly linked to their life-saving jobs. Without proteins, life as we know it wouldn't just be difficult, it would be impossible. Greetings. Ever notice how an egg transforms from runny to solid when heated? That's protein denaturation in action. Denaturation means the protein loses its natural or native structure without breaking the peptide bonds, so it's like unfolding a perfectly folded paper crane, not tearing it apart. This can happen due to heat, a shift in pH, or exposure to chemicals, leading to the protein unfolding and losing its biological function. For example, when cooking an egg, transparent albumin proteins turn white and coagulate, signaling denaturation. Importantly, the primary structure, the amino acid sequence, stays intact, but the secondary and tertiary folds are disrupted. Sometimes, if the denaturing agent is gently removed, proteins can refold and regain function, a process called renaturation, though it's not always guaranteed. Let's talk protein pH balance. Every protein has an isoelectric point, or pi, which is the special pH where it carries no net electric charge and becomes the least soluble in water. Basically, it doesn't want to play well with water molecules. Below this pi, proteins carry a positive charge, and above it, a negative charge. Think of it as proteins wearing an electric bracelet that changes from plus to minus depending on the pH parity. This property is super useful in labs for purifying proteins, especially through a technique called isoelectric focusing. At their isoelectric point, proteins tend to precipitate, meaning they clump out of solution, making separation from mixtures easier. Imagine a graph plotting protein solubility against pH, dipping down to a minimum clearly at the isoelectric point. This visual helps us predict when proteins will dissolve or drop out. Understanding pi is fundamental in biochemistry and pharmaceutical industries alike. Time for some number crunching. Suppose you have a polypeptide chain made up of 50 glycine molecules and you want to find its molecular weight. Each glycine has a molar mass of 75 grams per mole. But here's the catch. The formation of peptide bonds between amino acids removes water molecules, each weighing 18 grams per mole. Since 20 amino acids form 19 peptide bonds, for 50 residues, there are 49 peptide bonds, each subtracting a water molecule's mass. So, total mass equals 50 times 75 minus 49 times 18. Doing the math, 50 times 75 is 3,750 grams per mole, and 49 times 18 is 882 grams per mole. Subtracting 882 from 3,750 gives 2,868 grams per mole. Visualize a chain of glycine molecules linked by peptide bonds with water molecules popping off at each connection. That's protein polymerization in action. This calculation is essential to understanding protein size and behavior in biochemical studies. Let's wrap up today's protein journey. Remember, the peptide bond is the -CO-NH- link that strings amino acids together. The isoelectric point, or pi, is the pH level where a protein carries no net electrical charge. To find a polypeptide's molecular mass, use this formula: the number of amino acid residues times their average molar mass minus the number of peptide bonds times 18 grams per mole because that's the water lost during bond formation. Also, keep in mind the secondary structures. Those famous alpha helix and beta pleated sheets are where proteins start showing their elegant folds and patterns. Proteins truly are the molecular superheroes of life, folding and functioning in remarkable ways. That's it for today's deep dive. Chemistry is fun when you can laugh and learn, so keep exploring and see you in the next session.